Today's speaker is Chris Lovejoy. Chris has been with Certara for over 16 years. Originally trained as a pharmacist, his experience in the pharmaceutical industry includes PK/PD analysis, drug safety, and clinical drug management. Based in the United Kingdom, Chris has been involved in all the major European deployments of Phoenix Solutions, acting as a technical and project management lead. Chris, welcome to today's event. I'll now turn it over to him to begin the presentation.
My name's Chris, and I'll be leading you through this presentation, which is essentially a primer on CDISC SEND and how we may be able to help you with some solutions at Certara. So let's move on to the agenda. So today's agenda, I'm going to cover a few things. I'm going to provide an overview of SEND, so what is SEND and how it may affect you and your organization. I'll briefly go through some of the requirements of SEND. So the requirements of SEND are laid down in the SEND implementation guide provided by the CDISC organization. So I will go through some of the important features of SEND compliance with this webinar. And then I'm going to describe and demonstrate how Phoenix can support the generation of SEND data sets. And so we'll use a standard workflow, a typical PK tox workflow, and then we'll show you how we can use a workflow template and some plugins to provide SEND compliant output. And then, as Suzanne mentioned, at the end we'll have a chance for a question and answer session where you can pose some questions and we can discuss some of the things that I've shown you. Okay, so let us move on to the next slide. So the question on this slide is, what is SEND? And SEND is basically a data model developed by the CDISC organization. And the CDISC, well, what does CDISC stand for? CDISC stands for the Clinical Data Interchange Standard Consortium. And basically, it's a non-profit organization that's dedicated to provide sort of a platform independent data exchange standards. So as part of SEND, as part of CDISC, there is a SEND group that are involved in generating non-clinical data sets. And these data sets are really models for submission that allow data to be presented in a standard format, so that regulatory authorities such as the FDA and the Japan PMDA can receive these data and analyze them in a more efficient way. So that data standard now has become important because as from December 18th, 2016, the submission of electronic data will become mandatory. So as mentioned, the FDA and the Japanese PMDA from December 18th, 2016, if you're going to have a protocol signature on or after that date, you'll need to provide data in electronic format, and the electronic format for those data will be in the SEND format. So SEND stands for Standard Exchange of Non-Clinical Data. Okay, and it's basically it's the standard data tabulation model for non-clinical studies. So that is what SEND is in a nutshell. We'll go into a little bit deeper in the next few slides. And so we look at the next slide here. What are the benefits of using SEND? Well, the goals of SEND are, you know, as I said before, providing a standard for electronic data transfer, making it easier for organizations to share data. So, and those organizations may not be the regulatory authorities, may be organizations that, you know, standard sponsors deal with, like CROs or third-party data providers. But it becomes important because the FDA now want data to be submitted in this format as well. However, it does give other benefits and advantages if you use the SEND format. So it increases the efficiency of a submission because the regulatory authority now understand the data formats that are coming in. They can develop tools and programs that quickly can analyze and review the data. So, you know, the CDA, the Center for Drug Evaluation, pharmacologists and toxicologists can much easily now process the data and review a submission, and hopefully make the process a lot quicker for the sponsors. And then the other offshoot is because you're storing data in a standard format, sponsors or other organizations themselves can create data warehouses so that they can pool their data for all their studies in a standard way. That once you've pooled your data, you can then control it. You can check old versions. You can check changes in data and those kinds of things. And then you can also use sort of informatic tools to mine your data, so to do cross-study analysis, to create supersets. Okay, so now that's all well and good, but for the main drive at the moment is this December 18th deadline. So as I said before, if you have a protocol signature date on or after December 18th, 2016, and it's an NDA submission, you're going to have to provide the data in an electronic format specified in the SEND output format. So that is the main driver we're seeing at the moment for people asking about SEND. So let's move on to the next slide. Here's just a quick timetable, as I mentioned before, for electronic data submission. So as I mentioned, if you have an NDA, a NDA, or a BLA, you're going to have to provide electronic data if your signature date's on or after the 18th of December. There are some exceptions for BLAs, and there's some guidelines around that. If you see at the bottom there's some guidelines around the exceptions. But essentially, you need to plan for providing data in SEND format. If you're involved in an IND, then the deadline's a year later, so that's December 18th, 2017. Okay. Next slide. Right, so, yeah, SEND domains. What are SEND domains and what do they mean and how do they affect me and those kinds of things? So essentially, if you're doing an electronic submission, you need to be able to provide data in electronic format, but it's not one large data set that you provide. What you do is you break down your data into what they call domains, and a domain is a group of logically similar data. So I've given some examples here. In fact, there are up to 28 data sets or domains that you could provide as part of your electronic submission, but I've given you a kind of subset of some of those domains. And so you can see there's a demographic domain, there's a disposition, laboratory vital signs, pharmacokinetic concentration, and pharmacokinetic parameters domains. These could be the data sets that you would need to provide for your electronic submission. And obviously, the demographic domain would include demographic data, and the PC domain would include your your time concentration data that you use for your calculations for your pharmacokinetic parameters. Now, the domains are structured in such a way, or format in such a way, that actually you can join these domains together to make a superset. So if you needed to join, I don't know, demographic vital signs and pharmacokinetic concentrations together, there are certain keys within the format of that domains, or those domains, that allow you to create supersets of data. So a very powerful data standard. So you can understand now how a scientist could generate the data sets they need based on these 28 domains. We're going to actually concentrate on 2, maybe 3 domains as part of this discussion. So, if you're doing PK work, then essentially the 2 domains that would be of interest to you and providing to your submission group would probably be the pharmacokinetic concentration domain, the PC domain, and the pharmacokinetic parameters domain, the PP domains, which are essentially your derived parameters that come from here. Now, there might be an additional domain that you would need perhaps. So if you were doing sparse or composite sampling, you might need to create an additional domain called the pool def domain, which is essentially a list of animals that provided a single profile for your analysis. So it could be that you need to generate PC, PP, and pool def domain. We're not going to look at the other domains. It's possible that other parts of your organization, different groups, might provide the other domains for you as well. But your domains would need to be compliant to the CDISC standard so that in the end, when you do the entire electronic submission, your data works with the other domains as well. Okay. Oh, the other I guess thing that we need to think about domains when you submit the electronic data. At the moment, the FDA accepting the data sets in SAS transport files, so it's XPT format, not CSV or Excel, but SAS transport file formats, so XPT files. So we'll show you how we can do that as well. Okay, so right, so we're dealing with quite a few customers at the moment who are trying to generate these specific domains, and some of the feedback we've got is on this slide. So essentially, the thing to realize is that SEND is a new and evolving standard. It's at the moment the implementation guide is at 3.0. There is a 3.1 guide that will be coming out pretty soon. But I think at the moment the regulatory authorities will be expecting the data to be formatted in SEND implementation guide 3.0. So that's the standard that we would need to comply to. But to generate these sort of SEND domains, these PP, these PC, pool def domains, is kind of a time-consuming process. There's not many tools out there that can do that for you automatically. Sometimes it involves quite a lot of manual steps or a lot of programming if you've got a programming group. So it's not an easy thing to do, and it's not an easy thing to replicate over and over again and keep to a certain standard. So essentially the problem, I guess, is for scientists, it's really not a scientific problem that you have to deal with. It's more of an informatic problem. And so there's a lot of times where you'll be submitting, you're working with other groups such as your IT groups or your data management group or whatever. So there's those kinds of issues as well. There's sort of cross-organizational issues you have to deal with as well. And the other thing you have to realize is that for a sponsor, ultimately, provision of the electronic submission data is a sponsor's responsibility. Now, you can use third-party people such as CROs or other external bodies to help you create these SEND outputs. But in the end of the day, it's going to be a problem for the sponsor to deal with, and it's their ultimate responsibility. Now, if you have to send your data in electronic format and it's non-compliant, not fully compliant, then the FDA can return the application with an RTF, a refusal to file. I think that's going to be unlikely for initial submissions, but I think it's a possibility later on, if your electronic submission doesn't comply with the standards, then that's going to delay your application. That said, we are working with several organizations at the moment who are in the process of providing test submissions. So that's maybe something you can think about if you're trying to get into working out how to provide SEND data. Maybe you would need to try and do some test submissions to the regulatory bodies to see if it's going to be acceptable. Okay. So let's move on to the next slide. So what's the challenge? Well, the challenge is really to turn kind of scientific data that's kind of readable and understandable to a scientist or a group of people, trying to turn that into a format that is in SEND format, or SEND, it has the SEND formats applied to it. So you see a typical table here, easily readable. Unfortunately, if you send this as your electronic submissions, it will fail. And what you would need to do, you would need to convert those data. So let's move on to the next slide. To something like this, which is essentially the same data, but obviously converted to a machine-readable format and into a certain standard. So that is the real challenge. You have the data already, mostly, but it's not in the format that you can send electronically to the regulatory authorities. So, what are some of the features of SEND? Right, you can see in this table here that it's quite a controlled sort of format that you have to provide the data in. And if we look at some of the features of SEND, well, for a start, there's controlled terminology, right? So certainly for the PK parameters and also things like specimens, there's a controlled terminology, which means that you have to report PK parameters and specimen labels in a certain way. You can't just provide what comes straight from your LIMS machine or comes straight out of a Phoenix NCA object. It has to be converted into a controlled terminology. You also saw in the previous slide that the column headers are fixed. You can't create your own column headers. There are some mandatory and expected column headers, and they have to be in a certain format. And within that format, the field sizes are sometimes fixed as well. Another thing you have to do is you have to provide all dates in ISO 8601 date format, right? So I've given you a couple of examples here. So this value here is a date time. You can see it's the 15th of February 2015, but it's also got a timestamp in there as well, in hours, minutes, and seconds. And there's a specific format to the time and date, and there's a separator here to say where the time finishes, where the time begins and the date ends. And the second example is an example of a duration or an interval. So again, this is showing a half-hour duration or interval. All dates and times need to be provided in this particular format. And then finally, I mentioned before, if you're doing sparse analysis, then you might need to create or you will need to create an additional domain to list the pool IDs for the animals used for a profile. So those are just some of the constraints you have when you're producing SEND output. As I said before, if you go to the CDISC website, you will find the SEND implementation guide that will list all the requirements that you would need for generating SEND domain data. Okay, right, let's move on. Okay, so SEND, well, I'm going to show you how the SEND solution works, but what we're going to try and do is to show you how you can do your normal NCA analysis in Phoenix and then just add on a SEND workflow template that will allow you to map data from the TK analysis or the NTA analysis, map it straight into a send workflow template, and then almost automatically generate the required outputs in send format so that you can provide those files to your submission group. So it should show you that using these templates, you can save some time, you can reduce errors because the templates are kind of validated, and hopefully because we've tested these templates for you, then you can actually increase your compliance as well. So your kind of overhead in terms of generating these data sets is greatly reduced. Okay, so if I move on to the next slide, I'm just going to go straight into a demonstration of a send workflow. But before I do that, let's just talk a little bit about the normal sort of way things happen. And this is kind of what we think is the normal sort of data flow for analyzing data. So essentially, it's a case of retrieving your source data, pulling your source data. You can be pulling your source data from either Excel files or a database or some other data source, right? You're retrieving these data. You're transforming the data to give you sort of data sets that you can do analysis with. And then essentially, it's a case of exploring the data, getting a handle on the data, eyeballing the data, working out what the data looks like. Are there any outliers? Is how you expect, what the protocol is, and that kind of stuff. Once you're happy with that, you can do some visualizations and look for some trends, and then move on to the analysis of the data. When you create your plots and your graphs and your tables and your listings and that kind of stuff. And you might have to go through a few loops of that. You might spot some outliers in your output, and you go back and explore the data, et cetera, et cetera, and then report on those data. Sort of collate all your outputs into a particular document that you can create a normal report in. So, if I give you an example of that, let me just share up my screen for a second here. Okay, so I'm just going to pull up Phoenix now, so hopefully everyone can see Phoenix. This is a typical workflow, right, that we have in terms of just analyzing the data. So we have data coming in here. This is my data set. I could be pulling this in from, as I say, an Excel or a CSV file, but you can see there's non-clinical data coming into my Phoenix workflow, and I have kind of the data that I need to do my analysis. And then I have I've got here a typical serial tox template. And so in here, I'm mapping in the data from my time concentration data, and I've actually got a ratio sheet as well that I've mapped some data in. I'll do my data preparation. So in here, I might do some exclusions. I might do some changes to time concentration, that kind of stuff. I might merge some data, but I'm just basically prepping my data. So, and also maybe I'll provide some LLOQ rules as well. I might also exclude some data as well. Okay, so in here I might be, I don't know, removing outliers or removing my placebo or control data. That could be part of my setup in here. So I just remove certain types of data here. Well, in this case, I'm removing a particular animal from the profile, and then I'm just eyeballing the data. So I'm looking at my time concentration data here, and I create some graphs and some summaries and that kind of stuff, so I can understand my data. And then at this point, I do my NTA analysis, where I produce my final parameters. Okay, so these are my final parameters, and then I'll do some further calculations, and then that would be my final step. And then my final step, I suppose, would be to generate a Word document that has all these outputs in. So if I run this, okay, I just need to bring this here. You can see that all the output I've done for my analysis, I've put into a table that I can start putting as an appendix or part of my report for an internal group. But as I said before, none of this data is in SEND format. This is highly readable to a scientist or for peer review, but it's not useful for SEND output. So how do we get this sort of standard workflow, the standard outputs from this typical TK or tox workflow? How do we get that into SEND format? So the idea is that, as I said before, our solution is not to change anything with your normal TK workflow. Our solution is really to add an additional template, workflow template to your project that allows you to generate CDISC output. And this template, you can load in. So you would normally load in from this kind of template here. And you'd load in here. Essentially, you'll bring in a new workflow that has no data in it, and it's a case of applying data to this particular workflow template. So I've done that down here because it does take a little while to run, and you can see here that this workflow template is broken down into 4 sub-templates. First sub-template is called import data, and any sub-workflow that requires user input has, we normally label with this user label here. So you can see here that actually I only have one sub-workflow that requires user interaction. And all that user interaction is to map in data into this template, okay? Now, for this first piece here, we've mapped in essentially what's come in this data folder here. So this is our time concentration data that's come in, okay? We've also mapped in anything that we excluded. So remember I said I excluded a particular animal. This has the data that I excluded in there and the parameters I excluded. Now the other thing that we have to work out is how do we convert Win non-lin parameter names into CDISC compliant parameter names. And so we have a mapping here for matrix terms and PK parameter terms. And for that, we can use some additional sheets that are provided with the template up here. So if we look at these sheets here, in this non-clinical workflow worksheets, you can see we have a PK parameter rate. I'm looking in the wrong bit here. I need to look at the CDISC piece here. I have a PK parameter terms sheet here. Essentially, all this is a sheet that maps what comes out of Phoenix Win non-lin and what the equivalent term for CDISC would be. So it's essentially a lookup table, right? So we're providing what comes out of Phoenix, and we're converting it into these control terminology that's defined by the SEND implementation guide. And then additionally, we have a column here that just says which of those parameters do I want to report in my CDISC output, in my SEND output, and we have an additional column here that just says yes. So that's how I convert Win non-lin terms into CDISC terminology. We have a similar sheet here for matrix terms. So these are the actual terms required by SEND, but I don't know how your data is supplied, but whatever is supplied, you might not have the same terms provided to you. So this is a way you can actually map the terms provided in your data set to CDISC terms. And it's just a case down here of those mapping those terms in, right? So I've just mapped in those tissue terms. I've also mapped in the parameter terms, and then those are the only steps I would need to do. So I'm loading up my time concentration data. I'm loading in my final parameters data. I'm loading in my lookup sheets, and that really is all I need to do. The rest of these things, the rest of these steps, substeps, are automated. You generate the output automatically without any user intervention. So let's have a look at the PC domain. So we're taking in here, we're taking this data here, and we're converting it in several steps, I have to say. I mean, it's not a trivial set of steps that we do or process that we do. You can see there's multiple steps in here, but essentially we're converting the data using Phoenix objects into the required file for send. So here is the output that we have for send, right? So this is the PC domain, the pharmacokinetic concentration domain, and here is the send output that we generate directly from here. And you can see things like, well, you know, our dates are now in this ISO 8601 format, and then we have control terminology all the way through here as well. So, you know, there's some additional columns that we need to create and provide these key fields as well, so that we can join this data to other domains as well. Okay, so we do a similar thing with the PP domain. So we take, we open this up in here. Here are the final parameters that come in from Win, Non-Lin, NCA, and what we generate is the PP domain. And you can see here that the parameter names have been renamed from this long name in from Win, Non-Lin to this control name in Phoenix, and then we have exactly the same sort of key columns to begin with, so we can join the data up if we need to later on, and then we have dates in ISO formats. But we've done a whole bunch of other things as well. Now, if I was doing sparse study, then I'd also need to create this pool def domain as well. So this gets generated automatically, but it's not obviously required, because for this particular data set I'm using serial data. But you can see here we also generate automatically the pool def domain as well. So that's really all that's required to generate the CDISC output. Obviously, there are some required fields from this workflow that we need. So if there's some workflow fields that are missing, then we need to make sure they're in the workflow. But it's generally just a case of mapping the data from this analysis workflow into this send workflow. So the final thing would be to, I suppose, generate the files themselves. You can do this directly from the output itself. So if you look at the result file here, we can send this to, or we can export this to a XPT file. So if this was PP, then I would rename this as pp.xpt, and that's what would be required as part of my output. Now you can see now we are into standard Phoenix functionality. It's basically saying, oh, well, you're changing this into a SAS data set. Let's just check that everything's here, and you can rename variables and that kind of stuff. Hopefully, everything is as required. So these are the column names that are generated are the actual SAS labels that are needed for the transport files. So you could do that for the PP domain. You can do that for the PC domain. Some customers said, well, you know, you've come this far, you've generated all this stuff automatically. It's a shame that we have to do this manual step where we export the file manually. And so, in addition, we've also created a send export tool. So if you look on here, we created a little tool that sits in Phoenix that helps us export these domains as well. And so it's just a case of choosing the outputs that you want to export. So I'm going to choose the PC and PP domains, and then for the data sources, I can pick up this PC domain file, and I can also pick up this PP domain file here. And if I click next, then I get a kind of preview of the data that's coming through. And you notice here that actually the data is read-only. I can't change the data, it's only for review. there are a couple of fields here, or a couple of columns that I can insert data into. So this is the PC reason for exclusion and the reason for not done here. You can put these things in manually. So for I can put in here perhaps PC reason I don't know, hemolyzed or something like that. Hemolyzed and specimen lost or something like that. Okay, so notice when I put those in, then the PC stat column automatically gets updated as well, and the exclusion flag gets populated as well. So there is a little bit of manual changes that I can do in here. So if I'm not reporting a certain thing, I can actually put reasons in there if I haven't got it in my workflow. Similarly, I can do the same thing for my PP domain as well. So I can put some reasons in there. So I can put, I don't know, and again, the PP stat column gets populated as well. And then it's just a case of clicking on finish, and then off it goes. It'll go and actually create the XPT files for you automatically. Now it does a couple of things when it finishes this export. First of all, it creates these sets back into your data folder here. Puts the name of the domain file, and then it puts a date timestamp there, so that if you do it multiple times, you can check when you actually did the export. And then as part of the setup for this send export tool, I can tell it where to send these files directly to. So I've got an output directory here, and that could be on your local machine or it could be in a shared area. But also it will go and create these send outputs in a specific folder for you. And as you can see, if I can make this a bit bigger, they're directly in XPT format. So there's basically, it's gone straight from the workflow to an XPT file. And then you can obviously open this up in SAS and have a look at the data in SAS. So should be compliant for you as well. Okay, so I think that's the end of my demo. Let's go back to the slides if I can. Let's see, stop sharing. Right, so let's have a quick look. So I've shown you how what a typical workflow is, and basically how all we do is add an extra workflow that allows you to create the send data sets and then export them directly in the correct format. So I guess in conclusion, we can say that using Phoenix to create send output using standard Phoenix functionality increases output and efficiency. It allows you to create some best practices and some standardizations. And once you've done that, I think your QA group are a lot happier, because essentially you validate the workflow and not the output. So it becomes a lot easier for you to get your output created and accepted by the submission team. And then hopefully we've shown you how this is generic enough to use across multiple studies. So you're not one sub-workflow, one send sub-workflow can be used with a variety of different studies. Okay, so this slide really just shows some of the other types of workflow templates that we've done. Actually, I showed you the TK workflow. That was the first part of my demonstration using the TK workflow. So we've created toxicokinetic, serial, and sparse workflows, co-administration workflows. Those are for kind of non-clinical work, but we also I suppose we've done workflows for bile ducted, cannulated, and IVPO and portal vein type workflows. And then for clinical, we've done these kinds of workflows as well. We've generated these standard workflows for our customers as well. So that's just a real plug for other types of workflows that we've done, and you can use over and over again with your data sets. Okay. So I think that's really my part of the Q&A done. So we can move on to any questions. We got any questions, Suzanne, from
thanks,
the answer?
Thanks, Chris. Great presentation. We, again, as Chris says, we'd love to, for our audience, to ask your questions for Chris now. So it looks like we have our first question from our audience. Someone would like to know, for ANDAs, in the past, we've been fairly limited to submitting PK-based data. Will ANDAs still only be expected to submit similar data?
I don't. Do you mean I don't actually know? Without looking at the guidelines, I don't know. I suspect initially you're fine with submitting the data that you traditionally submit. But to be honest, I would need to check up to make sure on that. I don't. I'm not sure on that, to be honest.
We also have another question about the. There was one slide that showed the domains that implied that ANDAs would only be concerned with PK concentration and PK parameter data. Since ANDAs have been fairly limited to submitting PK-based files, will these be the only data expected for phase 1?
I think so, yes, absolutely. I think so. Again, hand on heart, I'd have to just check, but I'm not really an expert on what is required from the authorities. I would. Who is there? The ANDAs are they being submitted to the FDA? Because it's fairly simple to find out what the FDA require in their ANDAs submission. If there, I can address this later on, perhaps. But I would say initially that yes, you would only need to produce PK data.
Someone would like to know when was SEND released?
Well, yeah, it depends what you mean by so SEND. So SEND is part of CDISC. CDISC was formed in 1997. Now, in 2011, July 2011, in fact, there was the SEND implementation guideline. So it's abbreviated to SENDIG. And that version was 3.0, which was sent out to review. And you can pick that guideline up from the CDISC site. It's free. It's a PDF file that you can download. In 2011, December 2011, FDA CDA announced that they would accept the SEND implementation guide 3.0. And basically, they put in their guidelines that CDA strongly encourages IND sponsors and NDA applicants to consider the implementation and use of the data standards for SEND application. And then, as I've shown in the slides, December 18, 2016, that submission guidance will now become mandatory. So essentially, you would need to provide data in SENDIG 3.0 format, although there is a 3.1 document out for review as well, so that might become implemented in the near future, but at the moment it's 3.0.
Someone wants to know, when will new controlled terminology be required?
Right, that's another good question. So as I mentioned before, that's the SEND. The SEND standards is an evolving format, so it's continually evolving. The control terminology at the moment has been limited to what's in SENDIG 3.0. As I said, 3.1 coming out soon. There is always new terminology being added to the guidelines, but I think this says I think the guidelines say a reasonable time for these new control terminologies to be added. I suppose it's a difficulty is to know what's a reasonable timeline, right? But I think within a year, the new terminology would be a reasonable time. If there's new terminology come out, you should be compliant with that new terminology within a year, I would say.
How will the FDA use SEND files?
Oh, okay. So as I said, the FDA really I want to use send files for three things: for processing the data, for analyzing the data, and storage of the data. So the FDA themselves have a set of tools, Nonclinical information management system, and they're a suite of tools that are built to use send dataset information, and really allows them to process the data much quicker than if you were sending data in, I don't know, Excel or CSV or even PDF or printed submissions. So that's what they will first do. They will basically run that through their information management system, just to check if the data is good. It doesn't really check for the contents, it just checks the format is good and send compliant. And then they will run some validation checks against them to make sure that any variables all the variables are there, not missing, or don't have a supported value, or don't have the correct control terminology. And then once they've done that kind of initial validation, then they'll send it over to their toxicologists or pharmacologists and let them analyze the data. So that's how they'll use the files.
Someone would like to know how they can validate their send files.
Right, yes. So, yeah, there is a free tool available that actually we use internally here to validate our send output as well. So if I just share up my desktop here again, yeah. So if you go to this site here, Pinnacle 21net, there is a free tool. It used to be called the Open CDISC Validator. In fact, it's now changed its name for some reason to Pinnacle 21. But essentially, it's a free tool that allows you to run your data sets through this tool and give you a score to show you how fit your data is, and give you a validation of your data sets, right? So choose which data sets type you're sending, be it, this one's showing STDM, but there'll be a section for send as well in there, and then it will give you some information about the fitness for use, and hopefully you can check that your data complies to the send format using this free tool.
Someone would like to know where they can store their send files internally and whether you know of any commercial repositories.
Okay, that's a good question. So I don't know of any commercial repository that stores send files other than what we've developed ourselves. So there's a tool or a product called the Preclinical Safety Store, and essentially it's a set of Oracle tables that will store the 28 different domains generated for a send output or send submission. So we have a repository that will check in the data, will check the data as it comes in. It'll also have some functionality around nomenclature, so it will update nomenclature and that kind of stuff for you as well. And then the final thing, I guess the most useful thing, is we put a product called D360 on top of that. So D360 is a data visualization tool, data extraction tool. It basically you can query the database and ask questions around the data you see on there. So that's a tool we put on top of that repository as well to look at the data. I'm not sure of any. I'm sure there's some companies that have got their internal projects that store their data. I'm kind of aware of a company in Scandinavia that kind of did a homebrew, built their own system to pull in data specifically from CROs, actually, for pulling in data from CROs because they saw high variance in the data formats from different CROs. So it's a way of standardizing their CRO data and making them compliant, and then storing them into a single repository in-house. But apart from that, I don't really know of any other kind of tool or system to store send data.
Looks to be one final question from our audience. Someone would like to know, can this template be used to analyze PD data as well?
PD data. I haven't seen it used against PD. I'd have to look at your. You'd have to look at your PD data set and see what actually you're generating as part of your. Essentially, we can use any sort of data set as long as it has the required outputs needed for a send output, right? So as long as you've got the right fields there, they don't have to be in the right, they have the right column headers or even have the right format within them. But as long as you have the required columns needed for a send output, or we can derive the required outputs, then yes, essentially we can generate send data sets from that.
Okay.